Hello, everyone, and welcome to a very special Gen Live to celebrate, incredibly, the 40th anniversary of the publication of Genetic Engineering and Biotechnology News, the magazine covering the biotech industry. We're calling this episode Getting the Band Back Together, reflecting on biotech's baby steps and its march to maturity. I'm Kevin Davis, one of the editors with Gen. I'm joined by my colleagues, Chief Editor John Sterling and Senior Business Editor Alex Philippidis. Hello, gentlemen.
Hey, guys.
Kevin.
Guys.
And in a moment, I'll let John, who has been Gen's editor-in-chief for a mere 37 of those 40 years, introduce our three special guests today, biotech reporters, scientists, and old friends who've been covering the bioindustry since the very early 1980s. Each of our guests wrote some of their initial biotech articles for Gen, just as the industry and Gen itself were taking off.
This episode of Gen Live is brought to you by Isoplexis. We thank the team at Isoplexis, which just went public within the past 2 weeks, for their continued support of Gen Live. Isoplexis is a life science technology company building solutions to accelerate the development of curative medicines and personalized therapeutics. Their award-winning single-cell proteomics system reveals unique biological activity in small subsets of cells, allowing researchers to connect more directly to in vivo biology and develop more precise and personalized therapies. In a 2021 Nature Medicine study, Isoplexis's proteomics were used to examine immune cell activity in lymphoma patients that were receiving CAR T therapy that had cancers resistant to treatment or that had returned after remission. The study revealed that cytokine production by CAR T cells was an important indicator of their potency. This is just one of the many examples of Isoplexis's unique ability to provide predictive quality metrics. The Isoplexis platform fills a critical need in the field, accelerating next-generation therapies. Today's Gen Live is also brought to you by Gen Biotechnology, an exciting new peer-reviewed journal from Gen and Marianne Liebert Inc., publishing groundbreaking original research, insights, commentaries, and much more, launching in early 2022. More details, including submission guidelines, are at www.genbiotechjournal.com. And as usual, we invite your questions at any time. Please use the QA box. John, we're looking forward to hearing about the early days of biotech and Gen publishing with our guests. Over to you.
Thank you, Alex. As Kevin said, I've been privileged to be the editor-in-chief of Gen, to observe and cover the growth of the biotech industry since 1984. But today we're going to take a walk down memory lane and share some personal recollections from a few friends who were key members as freelance reporters in those early critical years. We'll hear how they handled breaking news from a nascent industry that was generating major scientific discoveries virtually every day, interacting with the stars of biotech, tapping into the mood of a young industry, and trying to get a handle on where the industry might be headed. It's a pleasure to welcome some old friends again, and here they are. Fred Gebhardt has been reporting and writing about the biotech and healthcare industries for over 30 years. He was one of Gen's first reporters covering the early evolution of the bioindustry involving human health and agriculture. Matthew Heil is currently Chief Scientific Officer of CureMar, where he's responsible for the oversight and direction of all scientific programs, drug formulation, and manufacturing. Matt trained as a research immunologist. In fact, he taught me, when I first started at Gen, some really basics about immunology, which I'll always be grateful for, and was captivated by the possibility of seeing new scientific discoveries translated into bedside medicine when the biotech revolution was just getting underway. Matt wrote for Gen describing these novel scientific discoveries from the perspective of someone literally on biotech's front lines. And Mary Jean Pramick is a professional medical and science writer with over 30 years experience in the biopharm and biotech arenas. In the 1980s, Mary Jean not only wrote regular columns for Gen, but she also did a monthly column called Bay Area Gene Beat. As many know, San Francisco was one of the hot centers of biotech back then, and she reported on news announcements and events coming from really one of the sizzling biotech hotspots. So everyone, thanks so much for joining us and being part of Gen's 40th anniversary. So let's start with Fred. Fred, we've all been involved with science as students, researchers, and journalists. How did the coming of biotechnology change how we looked at life science and medical research?
It was an eye-opener, not least because I had been living in West Africa with my wife as a Peace Corps volunteer and out of the country going on 3 years. And when I started for Gen, I was still trying to figure out things like barcode scanners, having more than one brand of toothpaste in the store. So suddenly jumping into the first annual seminar on biomedicine at Stanford was a new universe to me. I didn't even know there was a company called Genentech beyond the occasional mention in the San Francisco Chronicle business section. So it was a mind blower.
Matt, your recollections?
Oh, exactly what Fred was talking about. You know, all of this was happening all around us, and all of these names are household names: Genentech, Amgen. But it wasn't like these names were on the tip of our tongue. They were just starting. You heard little bits, you know, about the fantastic IPO at Genentech, and I happened to be involved with some things happening at Amgen. So it was like, wow, everything was exploding all at once. And it wasn't like it went forward in this orderly list of, now these are the people that you should follow.
How would you describe the atmosphere? You just mentioned Genentech beginning with their IPO in 1980 that would best characterize life science and biotech research, and why?
Well, I can give it from my point of view, because at that time, you know, 1980, I was just finishing up my doctoral studies. So you're really in the weeds. You know, I was studying immunology, and you're in all of these complex areas, very technical and everything like that. But what a lot of people don't know is that my original graduate training was in metaphysics and philosophy, continental philosophy, German idealism. And I had left that and gone to philosophy or gone to biology because I thought, like, well, this is all interesting, you know, but I want to do something in the world. Well, my experience in graduate school was, oh my God, I'm doing all really neat science, terribly interesting, but how does this get to people? It just seems so far away. I never touched a person. I only dealt with rats. And then Genentech and Amgen started to come out, and biotech started going. It's like, wait a minute, these people are like the guys down the hall from me, and they're doing stuff that's new for patients. That was the most exciting thing I ever heard in my life, is that because I was new to science, all of this complexity you're telling me can now be applied, you know, to somebody who has a disease that is so complex, cancer, you know. It was very, very hopeful. So I always thought it was very heady. It was like, wow, this is the way to go.
Fred, what did Genentech do to the industry, and what kind of feelings did it create?
I think they were probably more notable for things like their Friday beer bashes, at least originally. That's the sort of thing that attracted public attention, at least until they started producing products. And as soon as human growth hormone came and people realized there were actual things that could make a difference to real people's lives, suddenly it was a real company, a real world. Biotech was not a fantasy off somewhere on the side. It actually made a difference to people's lives. But it's part of paying attention.
Yeah. Hey, Fred, you were based in California back then, right?
In San Francisco.
Yeah. So in a previous discussion I had with Mary Jean, she described the companies, and she actually worked for a few of them, as the Wild West. Did you get any sense of that?
These days you talk about fake it until you make it. That's what biotech was doing 40 years ago. Essentially, nobody knew anything. It was, there were a lot of PhDs following their wild dreams, a few business people who actually tried to harness those dreams into a coherent business plan. But basically it was whatever you could dream up with. If you were convincing enough, you would find a backer and you would at least have a shot at making your riches, your fame, and hopefully helping somebody along the way. Big business came in eventually, but initially, yeah, Wild West is a good way to put it.
Yeah. Alex, you were going to ask me something?
Yeah, John, thanks. Tell us and the group and those watching about the genesis of Gen and your arrival at this publication.
Okay, well, despite what a lot of people think, I was not here at the birth of that wonderful magazine. It really was a result of Marianne Liebert. And, yes, she's my boss and my publisher, but I have to give her a plug here. She's one of the most creative people I have ever met in my life. She had been working in medical journals, and she had some when Gen started, but she sensed two things. One, there really needed to be a business publication. I don't mean business in the sense of just finances, looking more at the industrial aspects of biotech, what the other panelists were talking about from bench to bedside focus rather than just pure research. She saw that and she started Gen. And the other thing is, everybody back then was working on pretty much on the R&D, but she knew a lot of people, and a big issue back then was how are you going to make these things? These aren't chemical drugs. These involve living organisms, yeast, bacterial cells, mammalian cells. How are you going to make these things? Very, very complex. So if you, actually, let me see if I got it. I don't know if anybody can see. This is an early copy of Gen. You can see that. And it sort of showed, this is when we were a tabloid. Is this coming up?
No, it went into the background.
Okay. Well, you have to. You're going to have to trust me. Articles where there was a trade association started. The Chakrabarty case, who got the first patent on a living organism, was still kicking around in '81. Massachusetts General was getting into the business. The NIH panel. There's a gentleman named Dr. Klein who did a gene therapy experiment without approval, and he sort of paid the price for that. Congress evaluates biotech, and computer lingo works like the brain. So Gen was very early into what biotech has largely become now a data-driven industry and data interpretation. I actually came to Gen a year after I got out of the University of Minnesota, where I was a major in broadcast journalism. Back then I had a hell of a lot of blonde hair. I was a lot thinner. I looked pretty damn good on camera, but I couldn't get a job. I went up and down the East Coast. I went up to, down South Carolina, Virginia, and I saw this ad for Genetic Engineering News. And I said, gee, I got to do something in the meanwhile. This looks interesting. I covered some biotech in Minnesota. I came and had an interview with the then editor. We had a small office back then. Marianne Liebert had about 16 people. Gen had 3, the editor and associate editor and her husband, who did some work. So I was the 4th person. I said, look, I'm probably going to be here just a little while. You sure you want me? And he said, yeah, let's give this a chance. Well, 2 things happened. One, a month after I got there, that editor left. And Marianne surprised me when she said, do you think you could do this? And I said, no, Marianne, I'm here a month. I'm still learning the ins and outs of biotech. I didn't cover the degree that we do. And I think we need another editor, whom we hired. So I stayed on, and I really started to get interested in everything that was going on in biotech. I mean, every time they cloned a gene, it'd be a page 1 story. It's not like now where, deep sequencing and high-speed sequencing is the way to go. Virtually, as I said in my intro, there were new breaks every day in agriculture, in medicine, in diagnostics, even in environmental happenings. Bioremediation was starting to happen. And so I said, geez, it's a really interesting business. I'm going to stick around. And the editor who succeeded me left literally 1 year after previous editor did. Marianne approached me again and said, yeah, I think I can do this. And I said, but a couple of things we have to make. And again, I said, Marianne is so creative. We talked, and you couldn't see for whatever reason. Gen was largely black and white back then. And Mary and I were big fans of USA Today. Short, concise, a lot of color, tables, graphs, eye-catching. Not the old line journalism, although in a lot of ways I consider myself an old-fashioned journalist, how I approach stories. And I said, it's going to be more money. We're going to have to use more color, and we're going to have to up the rates for the big people. And Marianne agreed. And from that moment on, the magazine just took off with the industry. And as I said, I really liked what I was doing, working with Marianne, working with people like Fred and Mary Jean and Matt. It was just the most interesting thing I could possibly do. And slowly but surely.
John, I think we have Mary Jean by phone now, so let's bring her back in.
Wonderful. Mary Jean, are you back?
Oh, thank you.
Okay.
Yeah, we're having a storm off the coast here, so my reception is really bad. But yes, I'm in by phone finally. There we go.
All right. So talk about when you were working at those companies, just as they were emerging. What was it like?
Well, I found it very exciting because I'm a science nerd, and I just thought this was an incredible new, tack on science. Sometimes it was a little crazy at the companies. I won't name them, but we had to run looking for people who would conduct clinical trials. I remember jumping on a plane one day and flying down to down into Texas at MD Anderson. And I wasn't. I was just a consultant, but they just used everybody, who could help. Sometimes it got a little crazy. One of the companies I worked for, which I thought was an amazing thing, they had these huge tanks. They're 3-story tanks to build the monoclonal antibodies and the other kinds of the drugs they were making. One of the tanks got a virus infection, like we do, and we had to kill the whole tank with bleach and dump it because it was an infestation. I mean, it was like learning as we went, and they learned as they went. It's like this messenger RNA research that's been going on for 30 years was kind of quiet, but it kept going. It was just, I enjoyed it because I could go interview people and they talked to me. Scientists would talk to me and tell me about their research. And they were very, very excited about it. There were a lot of interesting personalities. I just re-researched Kerry Mullis. He was the surfer who ended up getting the Nobel Prize for PCR technology. I mean, things like that happened in those days. He recently died a couple of years ago, but it was, as Fred said, Wild West. Let's try it. So that was pretty exciting.
Yeah, I met Carrie a couple of times, and he was a real character. In fact, on the 20th anniversary of the discovery of PCR, I did a podcast with him. But Carrie being Carrie, and if you read his books, you know what I'm talking about. He didn't want to talk about PCR. He wanted to talk about why he thought that global warming was a myth. And he was.
I know. No, that's how he ended his life, as a denier of climate change and denying that HIV caused AIDS. He was a character.
Yeah,
but one of the other things, I don't know if you guys have covered it. Did you cover the Silmar conference on safety that happened, like 1980s?
No, that was before me. Were you there?
No, I wasn't, but I paid very much attention to it because I'm a basic lab scientist, and I was very impressed that the scientific world said, wait, let's go a little slower. We have to have some ethics about this. We can't just keep going because we don't know what this is going to unleash, and that was around 1980 as well. And, all of the scientists did attend with Herb Boyer and the other scientists from Stanford. It gave us time to pause and think, which I thought was a good idea at the time.
Go ahead. Mary Jean, or maybe Fred and Matthew can weigh in. Why is it that we've not been able to see something like a similar in recent years with the issue of CRISPR gene editing, raising ethical questions?
Well, and also the Chinese experiment in the in utero twins. I mean, sometimes scientists are going to do it. I don't know
think
myself
a lot of it
why we
is,
haven't seen it.
I think a lot of it is just politics has penetrated science like it has everything else. And it's much more a function of, I've got the right idea, I know what's right, and if you don't agree, then let's just shut you down and we're going to do it anyway. There's
Right.
no apparent reason to come to consensus anymore.
I think Fred is absolutely right. One of the things I've noticed that really changed in science with the advent of biotech is that these companies that were being formed, they needed money. And scientists took on a new role. They became very important in the raising of money. And results became the currency of Wall Street. And nowadays it progresses on a somewhat more rational basis. People announce results or trials, even though they do it by press release. In those days, they didn't know how to value a company. They had a function of how many PhDs you had, and each PhD was worth so many $100 or $50 million or whatever. People really did not know what to hang their hat on. And so all of a sudden you had a lot of famous scientists out there hyping results, really, making promises. And I think the amount of money that came to be involved in a fast-growing, fast-moving industry, generating millionaires every year, changed the nature of a more considered scientific community that Mary Jean is thinking about. That's sort of the community I grew up in. And I
Right.
too mourned the absence of a similar conference, because it's certainly something we need, but it's changed. This is a different age.
This sort of, your discussion just now sort of ties into another question I had, and this one again is for all of you. If you can talk about some of the opposition you might have encountered writing about research scientists suddenly setting up biotech companies and making considerable amounts of money, a result, as Matthew, you said, of the scientists becoming fundraisers. Were there not more people than we might have realized now who are uncomfortable with that commercialization of basic research back then?
Well, I can say what happened in the scientific community, and I didn't realize this at the time. It took a number of decades till I realized what the situation was. So when you're a graduate student and you study with a professor, you choose that professor very carefully. You choose them by name and reputation and school, and it's very purposeful that you go to study with them in that lab. And they become sort of, you become a creature of theirs. When you meet other scientists, they ask you, oh, where did you study? Oh, I studied in so-and-so's lab. Okay, I know immediately what you're an expert in, what you've probably published in, who your associates are. I know where you fit and what kind of expert you are. And senior scientists invest a lot of effort in creating people to go out and forward their ideas, their techniques, propagate their lab. And all of a sudden, these people are going off into industry like there's no tomorrow. And from the point of view of a major lab guy, you know, who had these big labs, lots of grants, this is a big loss. He invested, or she invested, in my case, 5 years in training me, plus the amount of time that the school invested, 2 years. So you're a big investment, and you're going where? And you're going to publish what? Because the word on the street was that you go with a company, your publishing is done. And if your publishing is done, your academic career is done. I only knew of one scientist who ever made it back from industry to academia. You could make it from academia, I was told. This is what I was told as I was graduating. You can make it from academia to industry, but don't ever think you're coming back.
How many people actually wanted to
And
come?
who was that scientist, Matt?
That was my advisory committee. Some of whom have
Okay.
passed on, and I do greatly honor my advisor. But she was very much in the traditional mold. She represented the traditional concerns of the traditional scientific community.
One of the interesting things I notice going forward from that is, particularly in practicing clinicians these days, every time you go to a conference, there are always sessions about how to protect your personal intellectual property and how to commercialize it. If you are
Right.
a surgeon, what are your surgical problems? What are your thoughts on a new device that will fix those problems? And it's a very accepted approach to commercialize whatever your work may be. And that's been a real change over the last 40 years.
Yeah, that's right.
Well,
Right. Everything is commercialization. And this is why I mourn the loss of the concept of Dr. Salk and his not patenting the polio vaccine. I mean, there should be a little higher bar here, but it's still a free for all.
yeah, well, I know in discussions both here and when I went to Europe over dinner, I was surprised at the number of basic researchers who really, in fact, one of the editors at Gen felt this way too, that this whole commercialization business was devaluing basic science. And the other thing was a number of them said, so many scientific discoveries are done serendipitously. And now everything was being directed for profit. And that's what businesses do. And a number of people did have problems with that. And the other thing that happened, and this was brought out by several of the people who wrote commentaries in our October issue, which I advise everybody to get a copy of or look at online, the amount of jealousy that was taking place. Thank you, Kevin. That's the book. And yeah, it's Lee Hood, it's Bill Hazeltine, and the MIT expert. They were all saying that as they were starting to make money, they were starting to not feel shunned, but they could just feel the jealousy. And at the point that Lee Hood, and I really recommend his article, that he had trouble when he developed his sequencer at Caltech, and he writes about this. I'm not saying anything's hidden. The president told him, we're an academic institution, we're not a business institution. You have to go out and if somebody wants to finance that. And I think he had to go to 16 companies before somebody did. And also he was an engineer in the bio department, and there was actually a move. And again, he writes about this. I'm not telling you anything, that to get him out of there, ostensibly for the reason that he was getting so much money for his new developments, and he eventually did leave. So there was this a little bit of jealousy and a little bit of pushback as the industry was still growing.
Well, I'll tell you, a good measure of this is many years ago, like back in the early 90s, I was friends with the guy who was head of the patent office at Columbia. Now, Columbia had the patents on gene, recombinant technology, big stuff. But I went into his office. This is Columbia, big time institution. And there were papers, everywhere. And he was essentially one guy in an office. And I spent the afternoon with him, and it was like help. He was being overwhelmed by stuff. Now, if you go by comparison to MIT's patenting office, that is a slick, and I'm not criticizing them, that is a slick, efficient way. They observe what their researchers are doing. They look at the publications. They get the information almost immediately out to the industry. They organize conferences between people. It's like what happened is universities realized that, we're looking down on these academics, but there's a lot of money to be made, because one of the first people who made a lot of money was Columbia. And that changed things.
Yeah, it's funny you say one man, Matt. I was invited to go on a tour in the East European country in the early 90s. And this one man not only handled patents, handled science reports and everything. And there was this rickety elevator you took up to the, I believe it was Hungary. And you took up to the 4th floor. And like you said, the floor was covered with papers, stuff was stacked up. And this one guy was lit. Now, again, Hungary was just getting started in biotech, but still one guy was handling an industry that was evolving at Star Trek pace.
Mhm.
That just stunned me as well. So that was one of the things.
Definitely.
Yep. Alex?
Yeah, we're talking about commercialization being an example of a controversy back in the day. What were some other controversies surrounding biotech research that you can remember either covering, writing about, or being involved in? And how many of these are still with us today?
Mary Jane?
Well, I will lean on the ethical issues. One of the basic ones was, are we creating life? Do we have the right to create life? Do we have the right to change genes? Who are we as human beings to do this? And that one has been kind of swept in the background, but it's still there. From what, I can hear it.
I think the objections were much more upfront in the agricultural biotech segment.
Oh, there you go. Right.
If you're talking human therapeutics, it's easy to put in a tagline, or at least the thought that we are fixing nature's errors. TNF inhibitors can fix an overactive immune system and deal with autoimmune disease. Human growth hormone can add in what nature forgot to give and created somebody who's too small. You don't really have that in agriculture because there's not the same sense of immediacy or need. It's much easier to talk about frankenfood with genetic modification than it is franken drugs, where you're bringing in a monoclonal antibody. There are 100 million people out there who are saying it may be franken, but it's saving my life.
Right.
Well,
however, in the food industry now, we've got the big pushback for organic foods. That's going to weigh. The controversy is still there with the GMOs. I think. It's actually getting bigger, and we may not be able to use them as much with climate change happening. So that,
or it may go the other way,
is
and somebody may figure out that if we can modify water uptake by plants, then we can extend growing areas. You depending on where you're coming from politically and ethically,
right?
You can argue it in whatever direction you want. And that kind of
I really agree that the ethics, and I think it's because the three of us, Fred and Mary Jean and myself, are coming from a similar time. The ethics of this was on everybody's mind. I remember a good friend of mine down the hall, she was graduating and she was going to go work for Eli Lilly, which was just starting a genetic engineering program. She went to her minister to ask if it was ethically permissible for her to work in genetic engineering at all. I didn't even know what the program was. And it really impressed me. It's like some people took this very, very seriously. I noticed, I think, a less important issue. Ethics to me was the big thing. Another smaller thing that I spent a lot of time dealing with is a tremendous amount of enthusiasm and misunderstanding about the speed at which the science would generate concrete products. Everybody thought that, that old thing, the time from bench to product is 10 years or 12 years or whatever it is they say. Everybody thought, oh, well, now it's going to be 10 months and 12 months. It probably cost me a couple $1 million, and nothing could be further from the truth. And I spent a lot of time telling finance people who did not understand these matters, I mean, it's not their area, that's not happening. We may get better cures, but we're going to pay for them in terms of time and money. And then in the late 90s, when finally people were realizing monoclonal antibodies were not curing everything under the sun, and it was hard, and they all wanted to abandon them, I had to put on a different record and say, like, no, this is still worthwhile. It's innovative. It can be newly effective, but it's not going to be quicker or less expensive. That was the issue I had a lot of trouble dealing with.
Well, one of the issues that I thought was mushrooming, and I've kind of followed it and attended the medical conferences lately, in the cloning the sequencing the human genome and getting the instrumentation faster and faster and faster and cutting down the cost. And now, we have everyone and their uncle checking their relatives with these DNA sequencing products. That went pretty fast. That technology did happen fairly quickly, but it's still, I think, I still want to see some more controls on some of these things.
Yeah. Well, in addition to GMOs, and again, this is back in the 70s after similar and 80s, maybe into the 90s, there's a big concern still with releasing unapproved recombinant organisms. Could they get free, say, in a field and incorporate themselves into other plants that weren't meant to be genetically engineered? And the two instances that I remember, and we covered them big time in Gen, somebody was working on frost-resistant strawberries. You put a gene in, I think it was the gene that you find in Arctic fish so they don't freeze. I'm going back now almost 30, 40, and they put that, the strawberries, and the guy was working on it, some academic, was so frustrated, and we can talk about this later because somebody asked me about, did I think this industry was going to become what it eventually became? And I said, yeah, but one of the things was regulatory affairs. Really dry stuff to write about, but critical, really critical. And this guy couldn't take it anymore. And somewhere in, I think, Oakland, California, he put the things out one night on his roof. Somebody found out about it and all hell broke loose. And then there was another scientist who was working on a rabies vaccine, and he apparently went down to South America without approval, as far as I remember, and started injecting foxes. And again, people got very upset there. So there's this real fear, 10, 15 years after, similar, not just about GMOs, but the release of not only unapproved recombinant organisms, but a lot of people still nervous about, and I hear some of that here right now, recombinant organisms. What will they do? And so that was big, and I'm sure it still is in some quarters. Hey, let's move up to early technologies, because Mary Jean, you talk about how fast sequencing has become. Talk about, in the early 80s, what were the technologies that you guys were writing about, and how many of them and which actually did you see transform biotech research? Why don't we start with you, Fred?
One of my first stories was about DNA sequencers, which in 1983, I knew what DNA was. I had minimal exposure, and okay, it's a chemical, it's a chain, there must be some way to put it together. And of course, it's a little more complicated than that. And it turns out that some of the first entrants into the space went broke because they kept putting out machinery that simply wouldn't work. And you finally got companies like Creative Biomolecules that put out devices that actually worked, and then they had to deal with users that couldn't follow instructions and would let the reagents crystallize, which scored the valves, and things fell apart, or they used the wrong solvent, dissolved the seals, and flooded the machinery. And you got a lot of problems where the science was good, the technology worked, but they didn't really have a user interface down. and they weren't user foolproof. And it was getting to the point, at least with the DNA sequencer, that for a lot of companies, unless you had a huge need, it was still cheaper to have you grad students do all this stuff manually than to buy into the new technology. And we're only talking machines that were $30,000, $40,000, which even in the early 80s was not a monstrous amount of budgets.
What's
I'm
that?
keen to ask, I'm keen to ask the panel. We touched on a couple of interesting personalities, including Carrie Mullis, speaking of PCR. But who are some of the other fascinating personalities, scientists, or executives that that were the pioneers back in the day that you enjoyed covering and interviewing?
Well, because I did a lot more writing in those days, and again, when I started, there was some big companies, and that's where a lot of the action was going on. And I found out that, as you said, Kevin, the company largely reflected the personality of the CEO or founder. And I'm thinking of Genzyme, run by Henry Termeer, who was a fascinating man. He always had time for you. I never remember calling him running this big business where he wouldn't talk to me on the phone or at a conference. We'd go aside and whatever. You know, when they started, they were working on diagnostic enzymes, which a lot of companies were working on. Therapeutics were also there, but everybody wanted to prove a lot of this technology is worth via diagnostics. And so he was working on diagnostic enzymes, then modified them for the use of human therapeutics. And of course, the company went on to develop and market an incredibly big story in 1991, Ceridase, to treat the rare disease, Gaucher disease. And the company still considers that, its most important project. But getting back to the personality, you're absolutely right, Kevin, every one of these guys back then had something to make them different. And the thing I liked about him, just the humanness the guy showed. And the president of Sanofi Genzyme hit it on the head when Henry died, calling him someone with the ability to forge an intense relationship with everyone he met. And he made everyone in the industry feel like he cared about them as an individual. And I heard nobody say anything different. Another guy who struck me as incredibly entrepreneurial was the late Hubert Schumacher, who I got to know really well. And again, he was one of the first companies working on diagnostic monoclonals. And he's also one of the first, I recall, to really start engaging in multiple partnerships with research institutions and marketing with other companies. So he combined really this, again, he was an entrepreneur, but a good businessman. A lot of diagnostics were based on monoclonals, but when he tried to get approval for a drug, Centoxin, to treat septic shock, for which I still don't believe there's any treatment. We've sort of given up on that in terms of covering in Gen. FDA didn't approve it, and the company went into a tailspin. And the gentleman that I actually shared a number of conference tables with was the late George Rathman, who founded Amgen. We used to call that one of Biotech's bellwethers companies, along with Amgen, along with Biogen and Genentech. The guy was a tall and stately guy, and as I said, he was called the father of biotech. The company developed numerous therapeutics. I mean, he went on to form Icos, maybe some others. So we were always doing stories on Amgen because they were just pumping stuff out. And the two key products were Epigen to treat anemia and Neupogen, which helps cancer patients receiving chemotherapy. And I just found him a first-class scientist and an entrepreneur. Hey, Matt, you really straddled both areas.
Well, it's funny that you mentioned George Rathman, because that's the name I was going to bring up. I had the occasion I was doing some things for Gen, and I got involved with people who were writing the annual report for Amgen. Now, Amgen was important in these days, but we're talking early on. It was so early on, I actually could call George Rathman on the phone, which today, much older,
Yes.
is sort of like dialing up God and saying like, so what's going on today?
Right.
You just would never do that. But I actually, in some things, and I'd called George Rathman, and you're absolutely right. He was a gentleman. He was a great scientist. And at the time, they were doing Epogen. I was an immunologist. I thought that was just the greatest, most interesting stuff going on. And I thought they were a great company. And he was very good about positioning, how they wanted to put it in cancer. He was a really insightful guy. He was able to be a scientist, but he also understood how we make something of this that's useful in the clinic. I really appreciated that. I liked him a lot.
Yeah.
Well, I just wanted to say one thing about the two initiators of Genentech, Herb Boyer and Bob Swanson. I mean, Herb was a premier scientist at UCSF, and Bob was a business person, and somehow their personalities matched each other, and then it kind of set the tone for the whole company, just from the very beginning. Because again, I will go back. They were both pretty ethical human beings, and I was working as a young consultant at Genentech when it first started, and it did set a tone. We've got to be honest. We're not. I mean, this was human growth hormone coming out before some other people at Genentech did nefarious things, but the two of them kind of did, create a company where people worked really hard and were fairly ethical.
And Fred,
Genentech. So.
I really like Mary Jean's talk about the importance of ethics very on, because I think historically people have lost this point. That was a major concern early on. It was the first or second topic anybody brought up, and it was very credible. As I noticed, as this industry, what happened for me is I moved from academia into this industry. And the part of it I was involved with was actually raising money for these new companies. And I met some people, that were way outside of a little immunologist in a research lab. That's not my world. I met what was his name? David Bleck, and who was a big financier for these things. I met a number of people. They could gather together money. The trick was raise 100 million, get a business plan, send it out, raise the money, do the IPO, and move on to the next one. And I remember once going into David Bleck's office, and the four of us, he didn't look at us once. He was so busy doing these things. I mean, clearly it had evolved into a big industry. As a scientist, I was really surprised at the transformation. It was like business was being stuck onto the framework of complex stuff, complex science, but complex ethics too.
yeah.
I think there was also a real effect as the commercial world bought into biotech. Roche buying into Genentech and eventually taking over, which certainly changed the way the company dealt with the world. Smaller companies like Calgene, which originated the Roundup resistance and developed the products and was absorbed by Monsanto. And overnight, it went from being able to pick up the phone and call the right person at the company and ask, what about XYZ, to Monsanto, and they basically didn't answer their phones.
Yeah. By the way, speaking of ethics, when I got to Jen to show Mary Ann's foresight again, Thomas Murray, who was working, I think, in Pennsylvania, was writing a monthly bioethics column. And again, take a look at the October issue. He did an article for
That's
it.
right.
And he eventually wound up as the president of the Hastings Center. And he's still and he testified before Congress on a lot of the ethical issues. And also David Bleck, Matt, I interviewed him, and you're absolutely right. He said some physical issues that he talked about, but his memory, and he started, I think,
Phenomenal.
Genetic Systems was one of the first therapeutic monoclonal companies. And he was, but just stuff that he remembered and the way he talked about it, I was just amazed at that. So yeah, I can't recommend, just for look, the issue is not about Jen. It's about people talking about a little bit about the past of biotech, but a lot about where it's going. And I can't recommend that issue enough. Everybody on the team and everybody involved did a great job.
John, we have less than 10 minutes left. We should probably, Alex has a question looking, I think, more forward looking. We should probably just sort of end on changing the horizon.
Speaking of the horizons, yes. Let's
Sure. When you look at the industry today, the investment, the companies, the successes, does anything surprise you about how far we've come in these last 40 years?
Matt, I'm going to give you that in a second. Let me take that first, because what I was saying before, this is again, understanding science, not medicine, but basic science being commercialized on a big scale. And the big issues were patents. We ran a regular patent column, regulatory affairs, and eventually, you put a lot of money into this. Is this damn thing really going to work? These are the things that people worried about. That's what the scientists could most control, because they were good scientists. But regarding the regulatory affairs and the patents, that stuff is still kicking around, is still part. But, and I think everybody can agree, we talked about some companies, we talked about some personalities. We knew this was going to become what it has become today. I just wasn't sure when it would happen, which is why I've been here 37 years. And it's still going on. Matt, you were in both areas. How do you look at that?
Yeah, I was surprised at the emergence of what I call science by press release. Gen was unique in that, and one of the reasons I wrote for Gen, and I did not write for a lot of other things, is because you guys took the time to get the science right. And there was an attempt to balance perspective. It wasn't hype. Gen was not a hype journal. But if you went out to some of the publications or the press releases that were being floated around out there, or the, what do they call those things, those prospectuses and red herrings before they do an IPO, oh my God, it's like, as a scientist, they just made me terrified. And I think the emergence of science by press release is one of the things we've inherited that I did not see. And I think it's been a shame, because one of the things that should not be done that way is science. It encourages too much misunderstanding on the part of the public. And that's not because the public is dumb. The public is not trained. So it's not their responsibility. I think we've lost a little bit of the critical venue over the years.
How much do preprints make that issue more aggravate that issue, Matt?
I don't think they do. Actually, I'm on the other side with that. I like seeing it if it's handled by the community. Like I'm used to them being, because if I'm in an area, I'll get preprints. That's great, because I can see what's going on. I can supply some comments. But unfortunately, those things are being leaked, and until the key for science is peer review and duplication. Until scientists review it and duplicate it, it just, in my book, is not real. So we're missing that a lot today. And that's why you see somebody saying, this does this to cholesterol. Oh no, cholesterol does the exact opposite. Oh, this is good for Alzheimer's. Oh no, it's terrible for Alzheimer's. It's because that's the nature of scientific research. It goes back and forth like that. It needs careful evaluations. I sort of like preprints. I like the internet. I like the availability. How they're making journals put more articles online? I just love that, because I belong in a company, and getting access to the information is hard. So that's been a big boon.
Right.
We're also seeing a real move, I think, to research with regulatory affairs and products in mind, much more than open research with more serendipitous results. Increasingly, when I did regulatory affairs, the whole mantra was start with the end in mind and design your initial research program with the products that you want to submit for approval. Know the regulatory pathway you're going to start, which is fine if you are a pharma or a medical device company and you actually have something in your hand that you can work with. But that same push to start with the end in mind keeps going further and further back to the basic science. And if you can't show a potential clinical use or application, maybe we're not going to fund this project. Keep looking for something with more commercial potential. And I think long term, that's going to bite everybody.
Yeah, definitely.
Yeah, that could be. I mean, I'm still working in the biopharma industry. I think there is some good basic research still being done at, research institutes, and they will find their way out, even if there is no commercial end in front of them. It's just because they work. It is basic research. The one thing that I'm impressed with in this development of the COVID-19 vaccines is more the cooperation among companies and academia in working together, because this was a really critical thing that needed to happen, because of the pandemic. So I know some scientists in San Francisco are really impressed with the cooperative nature that is now out there among scientists. So I think there's hope there somewhere.
Kevin, are we up against the hour?
We are. Maybe squeeze in one more question if you have it.
In quickly as possible, each of you, what do you think is going to be the big development in biotech over the next 10 years? Short and sweet.
Oh, geez.
Is anything in science
More companies.
Sweet?
Okay, CRISPRs.
I think it's going to be, unfortunately, and I do not support this, the use of CRISPR for the editing of the germline.
Okay. Boy, I'll tell you, we could go on another hour here with all of this, not only the past, but the future. But we do have to end it here. And I want to say thanks a lot to Fred, Matthew, and Mary Jane. Thanks to Alex and Kevin for tagging along, for asking some great questions, and to Isoplexis for their support, and to Gen Biotechnology, our new exciting peer-reviewed paper. And we'd invite you to submit a paper to them. And thanks to all of you for watching today. Gen Live will be back next month, when we'll be discussing advances in neuroscience. So everyone at Gen and our wonderful Gen Live production team, I'm John Sterling. Adios.
Great.
Thank you.
Great, John.
Thanks.
Thanks, everyone.